Welcome to the second lecture of genetics. I'll try to make this one much shorter than the part one of genetics. In this in part two lecture, we're going to look at inheritance, and we're going to look more specifically at chromosomes and how things are inherited on chromosomes, and how chromosomes can affect inheritance, how disorders can come about because of chromosomes and inheritance. So first thing we're talking real quick about is chromosomes. Okay, we've all seen this picture here, just like that. All right, you may see them like this, and they keep going like that, and you'll have them lined up in pairs. So like this is chromosome 1. You get one from your mother, one from your father. They're not linked here. Okay, they're not linked. It's not mitosis or meiosis. This could be This could be chromosome 2, and this could be chromosome 3. Again, mother, father, mother, father. You may see it as P for paternal, okay, maternal, and paternal. Now, when looking at our chromosomes, we have 23 sets, or 23 pairs. So this would happen 23 times. And when we look at let me clear the ink here. When we get to the 23rd chromosome, that's our sex chromosomes. Okay, you'll have your X and your Y. Now notice, and this is of the 23rd, and you get the Y from your father if you're a male, and an X from your mama. If you're a female, you would have 2 X's. Okay. Now notice that the Y is much smaller than the X. That's not my handwriting. That's actually true. The Y chromosome is smaller than the X, and the X actually has more genes because of that, and we're going to look at what that causes here next. So we're going to look at some X-linked disorders, or genes, because the Y chromosome doesn't carry a lot of the same genes that X chromosome does. There's some unique inheritance patterns. For example, we're going to look at color blindness here. Color blindness is a recessive disorder. So big C is normal, so that's normal. Little c is the disorder, or colorblind. Specifically, the common one is red-green color blindness, what we're looking at here. And it's found on the X chromosome. And so how do you solve this kind of problem? Well, let's say we're looking at a mom. We've got a mom here who is, 2 X's because it's a female, and then a dad. Now, it's not found on the Y chromosome, so there's not going to be anything there. You don't have to put a 0 there. I'm just showing you that. And let's say the dad has it, so he's got a little c. And the mom is heterozygous. Okay, so she doesn't have it, but she's a carrier. This is what we'd call a carrier. She's carrying it, but she doesn't show it because it's recessive. Okay, so she's a carrier. And we're going to cross them. And again, you go through and make your Punnett square after you figure out your gametes. And then you'd cross them here. And so this will actually tell you, and again, C is not the best choice, but it'll do for now, as long as you can tell what's lowercase and what's uppercase. Oops, if I could write a little C here. And this will actually tell you what gender, because it's X-linked, we need to look at gender as well, because it is gender associated, because men, if they have it, there's nothing on the Y, so they're automatically going to have the recessive disorder. You want to make sure you have the gender related chromosomes on here, and you write it in this way. Okay, you can't just do CC and C and cross them because you don't know what gender probabilities. Okay, so in this case, we're looking at this. We can say that 50% of females will have the probability. There's probability of 50% of the females being colorblind because there's 2 female possibilities here, and one of them is colorblind. On this side, you could say the same thing about males. It's a 50% chance that a male could be colorblind, and then there's a 50% chance of all the children that they could be colorblind. So that's how you would do a X-linked inheritance problem. Okay, and again, that has to do with the fact that the Y chromosome is shorter, doesn't carry as many genes. Another X-linked inheritance disorder is hemophilia. Okay, you don't clot blood very well. You just keep bleeding and bleeding and bleeding. So that's another type of X-linked inheritance. So make sure you understand X-linked inheritance, how to write it up, and what a carrier is as well. Next, we're going to look at autosomal disorders. Now, autosomal, so you had the 1 through 23 chromosomes. Your 23rd is your sex. The 1 through 22 are your autosomal chromosomes. Okay, autosomal chromosomes. So there's disorders that are found in those chromosomes as well. Okay, and so that's what we're going to look at. And there's autosomal dominant and autosomal recessive disorders. So an autosomal dominant disorder would mean if it's dominant, even if you have the normal. So let's say little h is normal, and big H is, let's say, Huntington's, because this is the truth here. It's not an example. This is true. Huntington is a dominant disorder. Okay. In this case, if you had this, even though you have the normal, you would have Huntington's because it's dominant. If it was recessive, let's say we were doing a recessive disorder, and little h was the disorder, we'll just call it disorder, and big H was normal. If you were big H, little h, you would be normal because normal is dominant. You would have to have both of them to get the disorder. Rather than in the dominant, you only need one to get the disorder. Okay, so that's the autosomal recessive and autosomal dominant. And those are not on the X or Y chromosome, they're on 1 through 22. Okay, examples of those would be like sickle cell. That's a example of an autosomal recessive disorder. And Huntington's is an example of autosomal dominant disorder. Next, we're going to look at family pedigrees, and I know you guys have to do an assignment on this. Family pedigrees is just a way experts, medical professionals, can track medical history. And what they do is you just set up a pedigree, and you look at whether someone's a carrier, they have it, or they don't have it. And there's a great example in the textbook on Figure 12.5. And basically, it just helps you track your family, and you can figure out the genotypes. And you would just use squares for male and circle for female. And so let's say I'm doing my mom and dad here, and I'm going to do blood type. Okay, I'm going to look at blood type. And let's say I know my dad's blood type A, so I'm going to give him an A there. And I know my mom's blood type B, so I'll put a B there. And I know they had me, and I'm male here, and I'm blood type O. So I'm going to have two of those, right? Because it's recessive. So now, based on this, I can actually figure out what is my mom's genotype. Well, it's going to be BO because I had to get an O from her. And I know my dad's going to be AO because I had to get an O from her. And then I can also track out and figure out my sisters and my brother. Okay. Now, if I was doing a disorder, I could say, well, if O is the disorder, I could say, well, she's a carrier, he's a carrier, and I would do halfway. If they have it, if we were saying O is a disorder, then I would color it all the way in. So that's how you do family pedigrees, and that's how they are able to track family history, looking at genetics and genotypes and phenotypes in that manner. So when you go to do your assignment, and the assignment lays it out there how to do it. This is an example, and there's an example in your book at Figure 12.5 to look at. Next, we're going to look at some chromosome errors or inheritance errors. So when our body is creating sperm or egg, and when we're going through fertilization, you can get errors within the chromosomes. And one example is polyploidy is what it's called. Polyploidy. Okay, and polyploidy means you have more than two sets of chromosomes. Okay, and in humans it would be fatal, and we don't really see polyploidy in humans, but many plants are actually polyploidy. So they'll have, for, let's say if we're looking at, like, we'll use humans as an example, right? We have 1 through 23, but for chromosome 1 there would be 3 different ones. For chromosome 2, 3 different ones. That would be polyploidy. Okay, so you have more than 2 sets of chromosomes. That's fatal for humans. You're never going to see it. Another example, sorry I jumped ahead there, let me clear this page, is aneuploidy. And this is more common, and this is actually causes disorders in humans. And it's a condition where you either have more or fewer chromosomes than normally exist in a full set. For example, we have 23 pairs, but which equals 46. But sometimes we can get 45 or 47, and that can cause issues in our bodies. For example, which we'll talk about this more in a minute, Down syndrome is caused by this. So, for example, if we have chromosome 1 here, we have 2, chromosome 2, we have 2, chromosome 3. All of a sudden, we have 3 for chromosome 3. During meiosis, the chromosomes didn't divide correctly, and we're stuck with 3 here, so our total would end up being 47. Or it could be the opposite. Maybe we only have 1 of 3, and then we get 2 here. So then you end up with 45 chromosomes. And that would cause problems as well, because we don't have a full set. You can also see this happen in the sex chromosomes, which we'll get to in a minute. Now, how does aneuploidy occur? It occurs due to non-disjunction, meaning the chromosomes don't separate correctly. Okay, when you're in meiosis, either the homologous chromosomes don't get pulled apart, or the sister chromatids don't get pulled apart. Okay, so you're not getting that separation, equal separation to all the cells during the creation of sperm or egg. So we'll see an example of that right here. So this diagram, and I'm going to straighten it out here, actually shows non-disjunction occurring at 2 different spots here. Okay, here you can see it occur during meiosis 1, where these are supposed to divide to different cells. They end up both going to the one side. They both go to this side, and then this side virtually gets deleted. And then at the end, you have too many chromosomes in those cells. And then on this side, it occurs during mitosis. So here you can see again in mitosis, you can see where it didn't divide correctly, and I didn't circle that very well right here. These 2 sister chromatids are supposed to, the sister chromatids are supposed to divide and go both ways, and instead they went that way only. So this cell right here will have more chromosomes than it's supposed to, and that's aneuploidy, and that can cause issues. For example, in Down syndrome, you get aneuploidy at the chromosome 21, so you end up getting 3 21 chromosomes, or 3 of chromosome 21, and that causes Down syndrome. Okay, this can also occur. So this causes Down syndrome. Sorry, I put this on here for you. Down syndrome. It can cause a variety of disorders. These are just some examples. It can happen in sex chromosomes as well, and you can get a variety of differences there. I'm not going to get into that, but you can get, just an X. You can get 3 X's. You can get XXY, which is where you get a lot of males that end up having more estrogen than they're supposed to, and can get some more female characteristics. Okay, so it can affect a variety of chromosomes. You can get a variety of disorders. They also have linked aneuploidy to cancer. There's still more research on that, but having extra chromosomes causes those cells to divide rapidly and go out of control and cause cancer as well. So that's one of the links behind cancer. The last thing we're going to look at here is what we call structural aberrations, or chromosomal aberrations, which just means there's some structural changes that aren't good that can be harmful in the chromosomes. The first one here is a deletion, and as you can see, part of the original chromosome has been deleted. Okay, and this can happen during the process. Sometimes things can break off, sometimes things gets pulled out, or during the copy and it just didn't get copied correctly. Here is inversion. What happens is, you can see this is the original here, and it gets inverted on the new chromosome. Okay. On the next one is translocation. This is translocation. I'll abbreviate, and you can find these in your textbook as well, figure 12.11. Okay, and in translocation, as you can see, the 2 different types of chromosomes swap DNA, and that's not good. Your DNA should be swapped between the same type of chromosomes, so you wouldn't want that to occur. Okay, and then finally duplication. Instead of having just one set, you have two of the same gene, and that's not good either. So that's duplication. And then this one was inversion. Okay, so make sure you just know the basics on those. I'm not super concerned that you're going to know specifics. We're going to get more into DNA replication in chapter 13 and 14, and actually look at more specifically some mutations or alterations that can occur during that. Just make sure you know these in general. And that ends the genetics part 2 lecture.